Okay.
So you are in Gurgaon, is it?
No, in Faridabad.
Faridabad, right. Okay. When did you join?
2013.
I see. Okay. We are really fortunate to have you with us today.
Thank you very much.
I was going through the profile. It was excellent research work. And
Thank you.
That's great to have you here.
No, it's my pleasure. It's
I
really
think, yeah, just a minute left. So you worked with Joydev sir, or?
Yeah, I did PhD with him.
Okay. So he is here with us. He visited earlier also.
Okay.
So
sorry.
It's great because we are bit location is quite remote, and
No, I would love to come there. I told him.
yeah, do visit, do visit. Otherwise, you will enjoy because it's scenic beauty as well as
yes.
Zero pollution.
Yes. Of course. Yes.
Yeah.
Food. So it is for 1.5 hours, I guess, no?
That's right. 10:30
that will also include discussion and all.
Yes, including everything. So I think we can start, Dr. Samrat.
I can start. I mean,
you can try sharing the slides. You can try.
So, should I start?
Yeah. How many participants we have now? 15. Record.
This meeting is being recorded.
So, good morning, everyone. We are very happy. Yesterday also, there were many participants joined online, as well as it was in blended mode yesterday, where our own PG and research students were present, and everyone benefited a lot. Today also, we have a very distinguished speaker with us. Let me formally introduce him. Ladies and gentlemen, today's our resource person is Dr. Samrat Chatterjee, and he is currently an associate professor in Translational Health Science and Technological Institute, Faridabad, India. He did his education from Calcutta University, Jadavpur University, and his work was on eco-epidemiological scenarios, deterministic and stochastic approach. And he has very good, to his credit, lot of journal publications in international journals of repute. His current interest is to understand the mechanism of biological phenomenon using mathematical and computational tools. And to his credit, he has 64 journal publications, 7 book chapters, and very recent publications in very good journals, including Journal of Theoretical Biology, Applied Mathematical Modeling, and so on and so forth. And he, apart from these credits, he has, I think, if I'm correct, around 6 patents, which are already approved. So with these words, I welcome Dr. Samrat, and I'm really happy, and I'm sure all the participants will benefit from his experience in this area. With these words, welcome, Dr. Samrat. You can go ahead.
Thank you, Professor Jamal. It's really nice introduction. No, patents, I think there's a copyrights, 3 copyrights,
Okay,
and
okay.
rest is fine.
I see.
So thank you, and I'm really happy and that I'm joining this. And I must let me share the slide. Slides are coming. Is my slide visible?
Yeah, it's visible. You can just enlarge it.
Is it okay?
Yeah.
Okay. So actually, the problem in the online is I can't see the audience. So it's only I can see my slides. So anyway, thank you, Professor Jamal, for inviting me to this, and I will thank my PhD supervisor, Dr. Joydev Chattopadhyay. Actually, he is the one who contacted me and asked me for this and give me this opportunity. So I really happy on this occasion. So I choose the topic. Actually, talk to him and. I choose the topic so that actually when I saw the program, it was mainly on the mathematical model on ecological and epidemiological part. So actually I did my PhD postdoc and start of my research career on that, but later I shifted to it's more on microbiology and human systems. So it's a field I have changed. So I asked him that how to go ahead. So he told that you present how mathematical modeling is helping in this kind of fields. So I choose the topic in this way, mathematical modeling and big data analysis. So I will try to merge 2 different fields in its own way, and how people are doing that. Because yesterday in the talk also, my sir, Joydev sir has told about the mathematical models and all, even in the inauguration session, that how different model Malthus and all comes out. And then Dean sir also told about this big data analytics. He talked about in the inauguration. So maybe this I hope my talk will give you some glimpse of how these 2 different fields are clubbed in today's research, and how we can take help of mathematical model to analyze this kind of big data and make some useful predictions. So you can see, actually, I can't see anybody. If anybody has any question at any time point, please let me know. So you can see from this title that we have this mathematical modeling and big data analysis. So 2 different parts are here. So I will first try to give some background of both this part, mathematical modeling, which you already have seen yesterday, but I will just go through very briefly. And some of the analysis, mainly the network analysis, which normally done on this big data. So to start with, so the mathematical modeling in biology, you all know, sorry. So you all know what is mathematical modeling, and especially if we talk about in the biology. So it is actually a useful tools that explains the the biological phenomena happening there. So you actually model a biological phenomena in terms of a mathematical language. You use it as a language, and then you solve that, and then you make your predictions or hypotheses or your findings, and then you translate it back to your biological terms. So more or less, this is how a mathematical modeling works in a biological field. So like in 1798, one of the first one to have this mathematical model by Thomas Malthus. So he made a prediction on this US population. So he made this a model, and he proposed this mathematical model which gives which works to the data of this US population. But of course, it has its problem, and later on logistic model was introduced, and it was used by Belgium mathematician Pierre Boulez in 1838. And he, because in the Malthus model, we'll see this exponentially growing. But of course, the population has its limit. You can call it a carrying capacity, and also he introduced that part. and it keeps on developing. Then these are on single models. So mainly in mathematical model, what you can find that we use these mathematical differential equation terms and we translate it and try to make prediction on the observations. So these are all single species model, and then later on it went to the two species model, which is famously known as Lotka-Volta model. So there are two species, one is prey and predator. So it was based on real data where they found that these populations were kind of oscillatory in nature, and then they made this model. Actually, two separate papers were made, one on this population, the other one on chemical this thing. And this Lotka-Volta model came into existence. And after that, a lot of models were made on this, and models were keep on making them complex. Then people introduce different forms of mathematical models. So they use delay differential equation. They make some delay in the time points, which is very natural in biological system, and try to understand what is the effect of this delay. Then they have the partial differential equation where they introduce space, changing the space along with the time, and try to understand it. Then stochastic differential equation, they use randomness, which is very common in a biological process because things are not as such deterministic. There are some kind of uncertainty exist in that system, and they introduce that. So these keep on making things complex, and as the model become complex, the analysis also become complex. People try to have new mathematical tools to analyze them. So in short, if I say mathematical biology, steps in mathematical biology, if you see, so it goes like that. So you have some observations, experiments, data, you take it from literature, you have your own lab determinacy, you talk to the biologist and take the data, then you translate that to a mathematical model in forms of some differential equations, ordinary differential equation, or any other form that best suit the data or the hypothesis behind that. Then you have your calculations, you estimate the parameters, you make all the prediction on that. So here when you make the mathematical model, you have to be very careful in two aspects. One is that it has to be realistic, at the same time it has to be simplistic, because you actually have to solve it at some point. Though we use a lot of computer now on, which I will show you, but for small scale model we prefer that we have some kind of mathematical analytical knowledge on that models. But then your model also need to be realistic, it can't be very unrealistic. So that is the challenge a modeler has to have. Then once you have the calculation on, you go to the simulation part, you make the new hypothesis and all, and then you suggest for new experiments which they do. And this is the way you can help ongoing experiments in biology. So now going to a different field, the big data network analysis. So those are mainly, I was talking about small scale mathematical models which we use using differential equations. Now with new age, we have big data everywhere, and not only in biology, in other sense. And in that case, one of the way to analyze the big data is the network analysis, where we normally use the graph theory of the mathematics. So there are many systems in nature which represents through this network. For example, simple, if you have the internet is a network of computers. So you have a router, you have these different computers, they are connected, and this is you can do it in a network analysis. Of course, the road network you have, where you can have these different roads, and they make a network of that. Air route network you have, and people use this network analysis in studying these things day to day. Then you have printed circuit boards are also can do that. Of course, the best one is the example of the Facebook, which is the best example of this network analysis which they have done. You have this website also is a network. So these are already existing there. Then people have shifted this to study biology. So in biology, the network analysis is everywhere, but mainly you can see it will available in the form of protein-protein interaction networks. So some of you might heard about this, but this is mainly actually in our body. What happens? The proteins normally interact with each other, and you can talk with each other through a kind of physical interaction in a sense. So these physical interactions made them, and they do it throughout the with many of the proteins, and this make them these networks. So you can see here, for example, I have given a schematic diagram that how they can come. So these are different portions where different kind of proteins can bind. I mean, depending on the site of a protein, the other protein binds. So even for a one protein, they can bind different proteins, not at one time, but on different times, which you can represent in a graph like this. So this is a famous called protein-protein interaction network graph. So this is an example, a graph from our lab only. So here you can see the proteins are actually represented in the circle, which we call a node, and the connection between the two proteins is called as the edge. So these are the edges we called, and these are the nodes which we talk about. So and here the size of the you can see the size of these nodes are
Sir,
different, the
excuse me, sir.
circles, and that is
Sir,
mainly telling about the importance of the
hello. Sir,
uh,
actually, we are not able to see the slides as you are talking on. So, we cannot see the slides.
okay.
Your slide is not moving, sir.
Dr. Samar, you are mute, I think. Please unmute.
Huh.
Yes, it should be okay now. Share again. Yeah.
Can you see my slides?
Yeah. Slides visible. Yes.
Is it moving?
Just try to change.
Yeah, I have changed.
Oh.
Is it moving?
No, not moving actually. Now it is moving. Yeah, this way.
It is moving like this.
Yeah, you should click here. No? Huh? What is this?
So in full screen mode, it is not moving.
Just go full screen again.
Okay.
Yeah, it is. It's not showing full screen and not moving also. Actually.
I'm not sure why. Okay, I just changing my.
This meeting is being recorded.
Is it coming now?
Yeah. Yes.
Okay, I'm just changing the slides.
Okay.
Is it working?
Not yet. I think in full mode it is not working. It seems. No?
So if I keep it,
When you click, it changes. Yes.
it is changing here.
Yes.
So can I use it like this or?
Like this. Go ahead. I think yes.
So is it visible or it is absolutely
Visible. Yes.
visible? Okay. Sorry for this. I don't know why it is happening.
Please go ahead. Yes.
So, okay. I will try my so that come. Okay. So I was talking about the protein-protein interaction network. So it's mainly the proteins which are interacting with each other. As I told you, it's the nodes are the proteins, and the interactions are these edges are their interactions. So this is the protein which I was talking about. So this is a network which we build in our lab. So these are these circles are the nodes. I mean, they are the different proteins, and they are the nodes we call it in mathematical terms, and they are connecting through these edges, which we call about the interactions, which we call as the edges. So other than protein-protein interaction network, we have metabolic network where so in our body metabolites are another biomolecule, so they also have their own reactions and interactions in that term. So this is the network built on that. There are networks in other biological systems also. So one is this ecological food web change. This is also they represent through a network. So having this saying these two things, one is on the mathematical model and one on the network analysis. So I will now merge that how they come together. But before going that, just I have this small story for you. So where this network analysis actually start with. So it all start with like a few years back. I mean, centuries back in 1738, 36, when the king of Kaliningrad, which in Russia now, I mean, has their city has actually 7 bridges. So he proposed a problem. He told that, okay, is it possible that if we start from any one of these bridge and crossing all the bridges once only, we can come back to the same possible. So is it possible or not? So people had did all the problems, and it was actually solved by Euler in 1736. And that was, people say that it's the first kind of its own graph theory where they did it. So he proved that he actually made that whole problem change into this network analysis, this network things, where these edges are the bridges, and these nodes which he denoted by the circles are the masses connected by these bridges. And he actually proved that the necessary condition for the existence of this desired root is that the number of bridges has to be even number. So if the number of bridges are even, then there is a chance that if you start from one point and if you cross all the bridges once, you will come back there. But since this here there are only 7 bridges, so he told that it is not possible to do that here. So this is people say this is a first of its problem in the graph theory which is start with. So anyway, as I telling you that two things I have shared with you. One is the mathematical modeling, which in details you already have known. The other one is the this network analysis, which we do in the big data. But now people are trying to use both of these. So if I talk about the work done in our lab, so it's mainly, here is a small overview of that thing. So you can see that we start with some experimental data, or we use literature, both, and it's in the big data we start with. Because human body, we have the informations of thousands of proteins and metabolites, we take them. Then we build our own large scale network. Then using that network, we identified some targets or some important pathways, and then we reduce it to possible small scale network, because in large scale network you can actually analyze the data, but actually if you really need to know the dynamics, then you have to take help of the mathematical model, the ODE based or the other differential equation based models. So we reduce it a small scale model, then we build mathematical models based on that small scale network, and then we make predictions, which we again go for the validation part. So if I talk about the research theme of my lab, so you can see it two ways. The first one, since I'm in a translational health science institute here, all the other faculties are from biology or clinical backgrounds. They are doctors, medical doctors, or they're from biology backgrounds. So they have a lot of data coming from clinics and other research from animal research or from cells. So we use those data, or even from literature. And in the first theme, we use all these tools of mathematical models and computer method to understand those disease mechanisms, and then try to identify some potential targets, which they go and validate. The other one is, we since in last 1 or 2 decades, especially in biology, with the development of this technology and all, there are huge data coming out. And in fact, now the available tools are getting shorter to understand this data. So there is a huge need, and people are working all over the world to have new computational methods and tools, or at least modify them in a better way, to understand, to get more information from this biological data. So that is the second thing which our lab work on. So that is more on mathematical part, where we develop tools and all, and then once we develop it, we try to use it on real scenario. So in today's talk, I will just go through both this kind of work which we do, and try to make you understand that how the different mathematical models can be used to dissect this kind of large-scale data. So the under the first team, I choose 2 problems, and the first problem was on the tuberculosis. So this is a work done here. So this tuberculosis is there for centuries. I mean how many years? And actually, the main reason is that they adapt human body very good. They use the human mechanism for their survival, and it's really become very difficult to actually eradicate them fully from here. So another problem with the MTB in the tuberculosis is that we now have different kind of drug resistance MTB, and this is because of problems by us only, that we don't complete our antibiotic and all, as the doctors tell you that you complete your antibiotic, because it makes you that bacterial resistance to particular drug. And so we now have multi drug resistant, total drug resistant, extremely drug resistant, this kind of MTBs are coming out. And so there is a huge need to have new targets for this. And the government is also saying by 2025 they have to eradicate this TB from India, and also people are working here. And one of the concept of this is that, can we manipulate human body, I mean our own immune system and all, so that with the existing drug it can be more effective. So this was one idea behind which this project was done, this work. To know that, to get that understanding that how we can manipulate human body, we first need to know how MTB is manipulating the human body. And one of the key portion is the manipulation of metabolites in our body. Because ultimately, if I go very briefly in the biology part, these metabolites are the sum of all your activities of a cell. Whatever happening in the gene level, whatever happening in the protein level, they all finally reflect to somewhere in the metabolites. So this was a study designed to understand that what this MTB is the bacteria which cause this tuberculosis, that what MTB is doing in the human metabolism. So this is the question which we frame here. So for that, what the experimental design was done, and the experiment done in our lab only. I mean, where I was there. So this experiment was done where they have taken different cells and they infected those cells with different strains of MTB, because one of the idea also was to understand this drug resistant MTB. So you can see here that this is an uninfected cell they have taken. Then they have infected them with this is one strain H30CRA. There are other strains like H37RB. This is GEL2283. These are clinical cell strains and they are drug resistant. These two. This is normal. And what they have done, they have used C13 glucose labeling something and used them in the mass spec. So the main idea of this experiment was that at every time point here written here 0 to 25, 15, 30 and 60 minutes, they try to understand that what the changes are happening at the metabolic level. Okay, so for that they have designed that particular experiment which I told you in very brief, and and they capture this in the human cell under the infection of different strains of the MTB. So they now have a data where the changes in the metabolites in the human cells are captured under different MTB strains. So this is, if I give a snapshot of this, so this is suppose the network of the metabolites. As I told you a few years back that there was a network, metabolic network. So this is a metabolic network of that. So if I give you a snapshot of that in different time points, you can see here that red are the reactions of the metabolites which have increases with respect to the uninfected, and green one was the one which get decreased, and the rest is remain same. So this we have only put for BND, one of the strain, just to give a snapshot. You can see that with different time points, these are changing. Now the whole thing comes up that what we get from this kind of data, because we need to understand that two things we need to understand from here, that how these things are changing over time and how they are changing over the strains. So this is the big data, you can say, in that sense, which I will show you. We have just used a part of this data for this study, but this was actually a big data of 3,000 metabolites and all. So how we can use the metabolic model? So the first one is so why we need a mathematical model. So first thing is that, so if I plot this glycolysis of different strains, you can see here that there is no particular pattern here. So if you take any big data, or if you start from there, you will try to identify things. You can of course find what is going up and what is going down, or you can take out the differences and all. But sometimes become very difficult to get any story or anything out of this, because it might give you some very random pattern and all. So here's the mathematical model can be very useful. So what we did here, so this is the only circuit we have taken for our model. This is not a very large scale data or large scale model, but medium, you can say. I've chosen this to start today's lecture, because then I will go to really large network, so you will capture there, because in that case I will not able to show you the models and all. So here we have taken a sufficiently large network, and this is a model which I made, simple model. It's just linear model in that sense, and we use this concept here. So as I told you that they use the C13. So what they have, I just give a little brief of the experiment, that how that changes to the mathematical model. So when they use C13, so normally the cell grow in C12. So they changes that to C13. So, and they see that how these changes are happening. So over the period of time, when they measure C12, it will go down, because you have removed C12 from the culture. And since you have put C13, over the period, the C13 will increases. Now, using these two, we can have, suppose this is a differential equation of a particular metabolites. So it will, this B, so it will control by the previous metabolites. So A will synthesis to B, and then again B will changes to C. And if the rates are R1, R2, suppose, and these are something coming from outside of this circuit, we kept it. So you can actually have a steady state concentration of B star on this one. And on this, we use this concept of, so the concept of the C12 and C13. So here you can see the R1 plus K1 in means this and this will give you, will get from the C12 and C13 combined thing from the data, and for the degradation, that is R2 and R out, we get only from say C12. Now all these things so what we need to know now, we have to use all this information to actually get R1 and R2 and all those parameters. so that we can actually see where these rate are changing and where these metabolites are changing. So we did that estimation, and we did all the mathematical things, and the final result, which I'm just presenting here, will look like something this. So all the metabolites which we got here, just to see that how we have estimated the parameters, this was the experimental concentration, and this is the base prediction concentration, we get it here. So once we have our model with the parameter set ready for that, that is showing a similar kind of trend as observed in the data, we now use some simulations to actually get that what is the key point that is making this kind of random things which we are observing here, this data. So what we did here, that for example, this graph, if you see this graph, so now we have models and the parameter sets for each of these strains and for each of these time point. So now we want to see that, okay, what is the point that is changing, for example, RV from uninfected. So if we try to change RV in a way that its metabolic profile come close to uninfected with the minimum number of changes in RV, then we can say that, okay, these could be the probably points where this metabolites, this MTB is working on, that is changing this RV to uninfected. So we did that here. So if we see that this straight line is the base point, and normally when you have uninfected, so here we have changed actually uninfected to RB, the other way around, that where the metabolites, where the MTB is working on an uninfected cell and changing it to RB. To capture that, you see that this is the these are the normal uninfected, which are different from this baseline. But when we make changes, actually I will show you, actually we just make changes in 2 points, 2 parameters. Only making the changes in 2 parameters, you can see all these uninfected changes towards the RV. And these same parameters were independent of the time point, I mean, and the strain also. The other way around, here we change RV to RA, because we want to see that whether these changes are strain dependent or time dependent or something. But you can say that fortunately and surprisingly, every time we're getting those 2 points. So if we change those 2 parameters in a particular profile, we can change that profile to any desired profile. And when we look for those 2 points, we found that those 2 points are actually DHAP synthesis and glucose uptake rates. So when we actually see the data, you find that all these reactions and metabolites are changes. But actually, if you go deeper and using this mathematical model, you can point out that which is the point which is making this difference. So everything you cannot target. When you are targeting to change a particular profile of a infected cell toward an uninfected or vice versa, you cannot target everything. But if you can identify a particular points where if you target and it changes to your desired state, then you can make that as a potential target. And here we got the same DHAP synthesis and glucose uptake. These 2 points, if we can target, we can change it to the desired states. And of course, then we look for some validation, and we did it on the glucose uptake rate. They did the biologists did the validation and found that yes, manipulating that glucose uptake rate, you can change the profile. We can further use the mathematical model, as I told you. So it's not only the mathematical model you can use on the big data to actually analyze those data, but you can actually use the mathematical model to predict further on the data. In case of this MTB, this fatty acid and cholesterol are very important. This is well known that this MTB use this cholesterol for its survival, use the host cholesterol system and all. So we try to understand that how we use that same mathematical model and try to predict that how this fatty acid and cholesterol changes over the strain and whether they are time dependent or not. And if you see it here, this is the prediction which we made from our model. So you can see, for example, in RA, this is less virulent. There is not much change. So this is a ratio between the particular strain and uninfected. So you see that there's not much change. But in case of other strains, for example, BND, you can see that there's a huge change in cholesterol, almost 10-fold, and fatty acid also. And over the time, the cholesterol, the change in the cholesterol decreases while fatty acid increases. And this pattern is actually different in gel and RV. So this time dependent pattern, which depends on the strain, we capture through our mathematical model. When we validate experimentally, we found that it actually matches there. So this kind of predictions also you can make using the mathematical models. So now we will go to a bigger data. So that is something where I have shown you the mathematical model which we made uh, to capture the changes in the metabolites and the reaction associated with it. Now we will actually use a much higher scale model, higher scale data, and for that we are using well known. These are well known as genome scale metabolic models. So the concept is more or less same, which I have shown you in the earlier example, but with a more complex system. And so here I will not go in the model part because it's more or less thing, but I will show that how these mathematical models is used in large scale data. So this is mainly, we are working on type 2 diabetes. So here also the type 2 diabetes, you all know that this insulin secretion from pancreatic beta cell decreases and all. But here we are trying to understand that in a type 2 diabetes, what is actually happening in the beta cell in terms of metabolites, because these these metabolic pathways actually finally lead to this insulin. So, and how can we target them to increase some kind of insulin secretion or what? For this, we actually use online data, and this is the data set we have used from here. It is on the gene expression data. So we are now using this genome scale model. They actually use this gene expression data, changes what is happening in the gene to and convert it to a metabolic changes, and try to see that what is actually happening at the metabolite level. So this is the model which we have used, and this is actually a clinical data set. So it was published where 10 non-diabetic and 10 diabetic patients were taken, and they all died. Most of them died due to this cardiac failure. And what they have taken is they have taken the pancreatic beta cells, and then they went for gene expression, and they provided that gene expression in that particular study. So we have taken that gene expression data from there, and we have used that in the genome scale metabolic model. So now you see that in the last example, I have shown you like 50-60 metabolites, and I have made that model. That was more indigenous way of making the model. But now the same concept we are turning to a much larger scale. So here the number of reactions are more than 7,000, number of metabolites are more than 2,500, number of genes are more than 2,000, and they are made using real biological data. So each of the genes are mapped with each of the metabolites and reactions and how they changes the metabolites in that sense. So in the genome scale metabolic model, for example, if I go here, so here this is the circuit which you can see. It looks like something like this, and here each of the reactions are provided. Some of the reactions are reversible and some of the reactions are irreversible. So that also are taken care of, and that actually are changes to a stoichiometric matrix where we actually use the reactions. For example, here this is an reversible reactions, and see A is going to B plus C. So here you find that here A, if I take the reaction 1, this is the reactions and then metabolite. So A is become -1 and B and C become 1 and 1. While in the second reaction, here you see that B is -1 while C is -2 because you have the 2 molecules and 1. So all of these reactions are changes to these, and with a flux. So here the assumption is that we analyze the whole thing taking the metabolites at the steady state values. So we use the whole thing equal to 0. If you know use this when you use the Jacobian matrix and all, the same way. Then of course we analyze these things, and since here another problem is that in this one the number of reactions and number of variables are different, and so it gives infinitely many solutions in that sense. So we use an objective function For example, suppose the biomass and all, to calculate the optimal solution from here. Though in this particular case, we have not used biomass, we use a different technique. So what we did, we actually we generate all possible solutions, and then once we taking this all the possible solutions, and once we use that in some kind of ATP synthesis and all, so the solutions which are giving same kind of ATP synthesis, we use them and we use the particular that ATP solution. But that you can choose. I mean, this objective function can be based on your biology or can be based on your need for the mathematics. So this is what the GSM model in very short. So once we have this model, we solve it. So there are different way we solve GSM. So I'm just giving a glimpse of what we can do here. So in here we have, as I told you, we have 10 control and 10 disease. For each of these control and disease, we make different GSM models. Then we did the flux balance analysis using efflux methods. So efflux methods, very shortly, if I tell you, this is something. See, in each of these reactions which I told you, so that can be controlled by one gene or more than one genes, and they can be in the AND or OR gate. I mean, one of the genes if act, it can work. In some of the cases, both the genes need to work. I mean, they make proteins which make complex, and after making the complex, they work there. So we have to use that AND and OR concept here. And also there are reactions which are controlled by the same genes, the other way around. That one gene can control many reactions. So all these possibilities we can consider in this efflux method. And then once we made that, we analyze the models. We had subject specific model. Then we use the sampling. We actually make them different on the flux states in the control and flux states in the disease. And then of course we go for the altered metabolic reactions. So sorry. So if we see here, this is the so after that we actually analyze the models to get different changes happening in the metabolic level and the pathways. I'm just providing some of the important results which we got here. So when we analyze the changes in the diabetic patients with the non-diabetic patients, we found that some of these pathways like this fatty acid synthesis and fatty acid oxidation, they are very high here in this case. And if we really go to the reactions, we observe that these two are playing a very major role. So now when we, so another thing which we got from here, if you see when we took the data, we saw that most of the patients died from the cardiovascular disease. So we are also looking at the what kind of changes happening in terms of the metabolites that are secreting from the pancreatic beta cell, or the metabolites which the pancreatic beta cells are taking in, because these metabolites, some of the metabolites can go out of your cell and come inside your cell. So we did that analysis, what are the changes happening in the metabolites, and we are more focused on, this first one, the metabolites that get increased in the diabetic patients and get secreted from the beta cell. So when we got that list of metabolites and we look for the literature, we found that there are 7 metabolites secreted by the beta cell which get increased in case of diabetic patients, are associated with cardiovascular disease. So this is one of the predictions or the hypothesis we made from this study, that because there was a connection between those patients who died of cardiovascular disease and diabetes. So we thought that this might be one of the linking point of these 7 metabolites. And we are now further, this is already published, but we are now working on this data, because if they are secreted from pancreatic beta cell, they must be found in the blood, their amount must be increased in the blood, and so on. The other conclusion, of course, on the different pathways inside the beta cell, and these pathways will actually link with the insulin secretion. And so what we are now doing, we are using all these pathways, as I showed you in the first slide, that from large scale data, when we get some important pathways or targets, we make small scale models. So we are now working on that, focusing particular on some pathway or some 2-3 metabolites to understand that what is happening there, which is showing these kind of significant changes in the large scale data. So these are some things which I told you, that we use some tools or models and all to understand data in terms of in real biological data, and give some hypothesis which the biologists can take for further. But in the other way around, there are also, we need to have new methods also to study this data, because as I told you, with growing this technology, even today there are lots of data coming with different shape and forms, the different experiments they are doing, and sometimes we feel that we might need more data, or we need to develop some data which a biologist or clinician can directly use it. So I will give an example of that we have made software which they can use directly. So the first example from this theme is on some concept node. So this was on the bistability and potential drug target. So before that, just I give you a background of what this thing is. That so this is on the cell signaling. So we all know that these are the cells, and all the cells have some, you can see some kind of antenna coming out of these cells. So they are actually basically receivers. I mean, the signal receivers from outside. So once they receive these signals through this network, which I told you, for example, the protein network, they go inside the nucleus to the and make using the genes, and they make the changes. And I'm just giving you briefly, and according the cell works. So this is normally cell signaling happening. That if suppose something is coming, you need to kill that some virus, bacteria. So that virus will come, the cell will recognize the virus, and then it will decide what to do. And this is how it works. But the problems come up if we have problems with the cell signaling. And most of the diseases are actually associated with the cell signaling, and some of the diseases are caused due to the cell signaling problem. For example, in case of cancer, autoimmunity, and diabetes, these are some of the very major diseases which are actually caused due to some cell signaling problem. Autoimmunity is something where our immune system start working which he is not supposed to do. There is no foreign things inside our body. He recognizes own thing as foreign, and start the immune process, which becomes then negative for our body. So this was one of the things which we were working on. We thought that, okay, if this is happening, then can we make some kind of tools or something where we use this kind of error signals to identify the targets, that where actually these error signals are happening. And in doing that, that was a long project which was done, and this is done in collaboration with my supervisor, Dr. Joydev Chattopadhyay. He's also part of this project. So I'm just focusing on something which I've recently published in Bioinformatics last year on the concept of bistability. So bistability is one of the signaling thing where I mean, mathematically we all know bistability. So there is two stable points, possible two stable point, and depending on your initial point, it goes to one of the stable point. But in case of biology also, it becomes very important, though it's not much work is done in terms of experiment because it's very difficult. But people think that many of the problems which are happening and can be solved by this bistability. Because, for example, there is a protein of GNK cascade which shows two functions. In one case, it's called apoptosis. Apoptosis means when the cell died. And the other sense, it's called cell proliferation. This means when the cell divide. So JNK, depending on its stability and which state it is, it can lead to a very different pathways. So what we tried here, we tried to understand that bistability concept through network motifs. And we tried to see that, okay, this cell signaling and this bistability, is it somehow related to the small network motifs which a protein is forming? So what we mean by network motif, so it is something like, if I say it's the smallest building block. Here I'm giving an example of 2 or 3 network motifs which keep on occurring in the network and which has some functional meaning. So for example, this is a feed forward loop, this is a feedback loop, and so on. So we are trying to see that whether this bistability, existence of this bistability has a relation with the choice of this motif structure. And if so, then can we actually use this motif structure uh, on which a particular protein is present in the cell, because a protein can be present in a cell in different motif structures. And if we can use that concept of this motif structure to identify which of these proteins are showing bistability and all, and use them as a target. So using motif structure, we translate it to proteins in terms of their occurrence in the bistability, and use that to identify any possible targets. So this is things which we are trying to understand from this study. So for this, we used a very simple motif. We only use uh, two uh, two node motifs, because even two node motifs, I will show you how it become complex. So, and any network can actually be reduced to two nodes. I mean, all the networks are actually combined, if you see, to finally a combination of two nodes, their edges, and how they are functionally linked with each other. And you can keep on adding nodes, and you can make the whole network. So here we are focusing on two nodes. So the aim is to capture if there are any existing association between this motif structure with the existence of bistability, which I told you. Second is that we are looking at that if there is any motif structure influence this input-output relation, because this is where we can make them target, in the sense that if any motif structure is has a more effect on this input-output relation, we can call it a sensitive nodes and so on. This I will show you in next slide. So this way we can actually divide and we can make them target. So this is what we also want to see from here. And of course, this we will show through this bistability switching through hysteresis. And finally, all these things will be used to make concept of identifying potential target, and then we use that with the existing data. And finally, we will see that these all things we will do in a deterministic way, but of course in a body it's not actually happening in a deterministic way. So we'll see that how stochastic these random perturbations are affecting this maintenance of this bistability, or whether this bistability can be maintained under this random perturbation and up to what level. So this is the very basic structure. This is the two node general motif. So let me just explain this one to you so you'll understand. So you take any two proteins. So these are the A and B are this to be a protein, and they are actually linked with each other. They can be linked with each other in these different forms. This is it's not that all of them always exist. I'm just putting a general one, that this is all the possible way they can exist. What is this? A can actually activate B, A can inhibit B. So this is the arrow where A can do both the things, for example. B can also activate A and B can also inhibit A. So this can also occur simultaneously. A can activate himself. This arrow is showing, and B can also activate itself. And the whole process will be governed by some input coming from outside. So this is just two nodes we have taken from a flow of information. So suppose there are 10 proteins in that line, then how each of these proteins is affecting its consecutive protein, this is showing that. So some information is coming from the upstream proteins, then it is affecting A can affect B in activation inhibition form. In reversal cases, B will also affect A, and there are some self loop where A and B are controlling. In doing that, we actually found that almost like 40 structures were there, which is forming in different forms, which exist biologically. But then when we look for the bistability, that which of the structures are actually showing bistability, and this we did numerically, basically. And when we went for like 10 lakh parameter sample set, we found that 7 of them are showing it. We are not saying the others, we really don't know, but these 7s are showing much more frequently bistability. So these are the 7 structures which we now take forward out of those all the structures which were showing bistability there. The next thing which we'll work on here is on the sensitivity of the parameters. That okay, these are the different topologies, these are the different structures, and these are all the parameters here. And we want to see that which of these parameters are sensitive in terms of the bistability for each of these structures. So once we have knowledge on the sensitive parameters there, then we actually try to understand that how these sensitive parameters are dependent on the choice of these topologies. So here you can find that this is the robustness analysis we did on these sensitive parameters. So in the y-axis, you will find these are the sensitive parameters which we get from our last sensitive analysis. Now, when these parameter values are varied from their uh, base positions, which is the starting position which we consider for our sensitive analysis, which we varied fold 2.5 fold both ways, we find that, and it is done for 1,000 times, so there is also a standard deviation here. So you can find that in some cases these parameters are, so these bars are the showing that how much they are varied and still the bistability maintained. So if your bar is smaller, for example here, that means if you vary those particular parameter, this system become unstable, monostable or unstable both. I mean, they lose bistability. But if these bar are larger, that means up to here they are showing bistability. So we actually see for each of these parameters in each of this topology that how they are changing in each, they are affecting in each of these topologies in maintaining the bistability. I will show how they are useful. I will show you in the next slide. So okay, I'll just go back. So here we found that some of the parameters are, of course, making bistability is keeping bistability for a larger range. In some of the parameters is for a smaller range, but it is also depend on the topology. In some cases, the same parameter is showing robustness for a large variation, where in the other topology it is showing for the low variation. So we just take this information further and try to understand that what it actually means. For example, here I'm just showing you one of the parameter sets and its effect on what we mean by this robustness sensitivity in that sense, in terms of maintaining bistability. So we have taken a particular topology and we have varied K2. So let me first show you the graph, what we mean by. So this is graph is called the hysteresis, which we plot here. So there are two responses you will get. So this is, we call it a reversible hysteresis, and this is the irreversible hysteresis. So first, let me tell you the how what we mean by reversible hysteresis and irreversible hysteresis. So here, if we increase the input stimulus in the system, so it is going to a particular state. So these blue lines are the two steady states maintaining here. So for a particular range, you will find that they are bistable. Outside that particular range, they are monostable or unstable. So so if we have if we increase the input stimulus, in one case this stability, once it's going from here, it jumps to the other steady state and it remain there. But the thing is that when we decreases the input stimulus, you will find that in this case, in the first case, it will come back to its original one because this is not going back here. So at some point it will come back here. But in this case, since this has going back to here up to I equal to 0, so once it moves from this steady state to this steady state, and even after that, if we decrease input signal, it will not come back to its original steady state and it will remain there. So this is the two concepts we have used in our bistability to identify the drug targets. So we said that the reversible hysteresis, the structure with larger reversible hysteresis could be a better drug target, because in that case, there is a chance that if we target it and reduce the, for example, the input stimulus or something, then there is a chance that it can come back to its original steady state. Here, assuming like the original steady state is your good steady state or your homeostasis steady state, and the other steady state is occurring because of some disease condition. So the condition where you can actually bring it back to its original steady state, we propose it as a better drug target. And here, this is exactly why I showed you the last slide, that how the parameters were measured. So here we found that this feedback loop was something which is controlling this reversible steady state and irreversible steady state in a very high way. So we used that. Then what we did here, so once we have this hypothesis, we really want to see that how we can use all this concept in a real disease condition or real state. So what we did, we have taken 3 cancer networks, breast cancer, ovarian cancer, and pancreatic cancer from literature. And then we build, we take all the proteins, whatever the protein changes happening, we have that data. Then we use that to build protein-protein interaction network, which I told you, but functional network. As I told you that 2 proteins can activate, can inhibit, and all those things come under the functional network. So we use a functional network for each of these disease state. Now in each of these disease state, we use that functional network to identify proteins which are present in these 7 motifs, which I shared, that those 7 proteins which are showing bistability. Now we see that how many proteins are present in how many of these 7 structures, and we rank them accordingly. That means more proteins are present in more of these 7 structures, that chances are high that that will show a bistability. And of course, in that case, we use the reversible hysteresis, because we know for each of these, I'm not showing you the figure here, but we have actually identified for each of these topology that constitutes the reversible and irreversible range, and we actually have ranked the topology based on the reversible hysteresis. So now we actually connect those proteins based on its appearance in each of these motifs and rank them, and we say that those proteins which are present in the most of the reversible bistability positions could be a good target. And this is the rank which we made. If you see the x-axis, this is the proteins which we rank here. Now what we found, and this is these colors showing that how many times they are present in a bistable region, and this is the rank which we made here based on the topology. So here you can see that under bracket we have written, and for each of these proteins, we actually went back to literature and find out that how many targets are actually drugs are targeting these proteins, are actually targeting them. So we have taken it from drug bank database, and in bracket we have put these numbers. So in whole, this picture is showing that the number of drugs that are targeting a particular proteins are more on those proteins which are actually showing reversible hysteresis, which is actually validating our hypothesis that if a protein which is showing reversible hysteresis could be a good target. And this is exactly what we observe in the literature when we look for the when we compare with the drug targets. And of course, in the last, we try to see that what will happen if we introduce randomness. So I'm not showing you the model. Here we have used the stochastic differential equation to get it to understand its effect on the bistability. And what you observe here, you can see, so when the randomness is small, you find that this is the true steady state. Okay, when we start increasing this intensity of the randomness, this steady state start increasing. I mean, the clouds start broader, and in when it crosses some steady state, one of these crosses some threshold value of this noise intensity, one of the steady state actually collide with the other one. So in nutshell, what I can say that when we increase this stochasticity in the system, it maintains bistability to a point, and all the theorems which you have considered is happening. But once it crosses that threshold, this noise intensity, this bistable becomes a monostable, and monostable in the sense that it goes to one of these steady state. So in that sense, it will become much more difficult to target it. So if the system becomes very noisy, all this concept of targeting this bistable node will become very difficult, because now they will not show all these reversible hysteresis and all those cases. And whatever the target we have proposed will only work to a certain amount of noise. So this is a small observation which we did at the end, that because the system which we are working on are not deterministic in that sense, there will always be noise. So the target which you are making is possible to a certain level, but with the increase in noise, which will of course will become more effective with a more acute disease and all, it becomes difficult to target them. So these are some of the conclusions which we made from here. So this is a tool which we made based on the concept of bistability to identify drug targets. So same thing now we are using in different actual biological scenarios, that wherever the bistability is existing, that what is the thing that we can target there. And also we are trying to understand the mechanistic understanding of, because we know that this is happening, but we need to know why this is happening, in the sense that why bistability or hysteresis are making it more target friendly. So this is something which we're working on, but we see from here that we can use a concept of mathematical concept to have a new tool to get more knowledge from this biological data. The last one was something is more, it's not mathematical in the sense, but it's more a tool which we develop for the biologists and clinicians. So whatever I have showed you has some advanced level of mathematics, and also we can sit with the biologists or clinicians and we can solve them. But there are many things which they can do themselves, in the sense that if we can have a very useful and user-friendly software where they can put their data, and the preliminary analysis can be done easily on that. So that was one of the concept of making this. So here what we have done, this was launched last year in 15th August by our director. So this is available in our website. This is called Connect to Prot. This has been copyrighted, and this is now been sent for publication also. So you can directly also go to this website using this the link. So what we have there in the particular Connect to Prot, we have in the background, we have a database of all the proteins, their interactions, where they are present in the cell, what are the disease they are affecting, what are the pathways they are affecting, any drugs available on them, what is the structure of the proteins they have. So all this data we have kept in the background. And then we also have made some algorithms to solve those network. These algorithms are already available. This is not the new this is not new method we have developed, but the algorithms which we have taken it from different literature, because here also we don't want to make it too complex for the clinicians and the biologists. We just want to make a software which they already know, that because these are some basic analysis which they already know. So we use those in this software. So mainly, what is the background of this software? So actually, there are few things which you need to understand to study a PPI network. For example, so directionality. Like in the last case, I showed you that we have used a directionality to know that whether protein A is affecting protein, inhibiting or activating protein B, and so on. So that is what we mean by directional informations. Then you need a pathway information, subcellular information. This all we need, and there are a lot of papers where people have shown the importance of all these informations. But unfortunately, till date today, these are the major databases available in the internet, and these are the primary databases, and there are other secondary databases which using the same database. So, but unfortunately, none of the databases has all these informations. So when you look for a particular information, you have to use 2-3 databases, and sometimes the problem with the databases is that the formats are very different. So you need to actually change them and move to the other database and all. And there is also very less database which actually have analysis part also. So they only have the database. You put your query, and they will give you some of the basic things. So keeping these two things in our mind, we made this connect to plot. And this was another DBT project which completed last year. So this was where we have put all the possible informations from different database sources and also from literature, which is not available in databases. We curated few from literatures like hallmark of the diseases. We put all those in here, and then we also added that analysis part which I told you. And this is the salient features of the connect to plot. You have directional information, you have multiple search mode, you have pathway information, you have disease information, all this, and then your localization, and these are not available in one single database as of now, and the analysis part. And this is the flow chart how it works. So you put a protein inquiry here. I'm just very briefly I'm telling you protein inquiry here. We have some background databases which will help you to build a protein network. Now, if you want to filter out something like I want a network for example, for mitochondria or for cytoplasm or for nucleus, because these are different part of the cells. So you want that, okay, no, I want a particular network for a particular compartment from the cell, or you want that, no, I want a network which has to do a particular disease or particular pathway. So that you can also do, and you can refine that network. Then you suppose want that, okay, I have a particular protein which I want to know, but I want to know the structure of the protein. I want to know that what are the drugs available on the protein. That also is available from here. Then you want to have more analysis on that protein. That, okay, I want to have enrichment analysis that says that what are the particular pathway it is enriching and all. Or if you want an influential spreader. So this is a algorithm where we actually find the nodes in a network that is the most significantly influencing the surrounding proteins. So it has an algorithm there, which this is published by somebody. We have just used that one here because this is easier for a biologist to capture that influential protein from here. So and then of course the other information like hallmark and this we have taken from literature. So if you go to the site, it will look something like this. Here you put the protein list, you will get the list here as I told you. Then you want some local properties, so you click here on any protein, so it will talk about that particular protein. If you want a global properties of the whole network, you click here and then it gives the global properties of this. And based on all this information, it is up to the user that how you use it. We are not saying that these particular proteins are targets or these particular proteins are trigger or they are just a particular protein. It's based on that what is it based on what is your requirement for that particular protein. What is your hypothesis or what is your question? So for example, just to show you, we have used this on a Chinese medicine. So that Chinese medicine is particularly used to cure breast cancer. So people have used that particular medicine, but they don't know that what are the targets of that medicine. So we have used the gene expression data, the proteins from there, and we put it in our network. And through all this analysis with this information which our connect to plot is giving, like these are the statistics, these are the hallmarks, and these are the spreaders which you get. We found that KTP said that there are 15 influencer spreader associated with this particular disease. So when you use this particular disease, there are 15 proteins which are seems to be get affected or could be targeted by them. So when we go back to the literature, we found that out of those 15s, 5 are actually known to be reported to be successfully targeted against the breast cancer. So we really don't know if they are target or not, but we have used connect to plot. We have shown this in the paper that in this particular case, we have used connect to plot to get an idea on the possible targets which these medicines might have shown. This can be done in the other way around. If you have a disease profile, you are putting it down, it might help you to identify or give a idea on the triggers of that particular disease. So this depends on the user. So connect to plot is not making any conclusion on this, but it depends on your query, it will give you the answer. So that brings the end of today's talk. So I thanks my teammate because this is only because of them it's possible. I am just presenting all these works are done by them. And of course, as I told you, I'm also one of the one of the project. 2-3 works at least on them, and future I get a guidance from Professor Joydev Chattopadhyay. He's keep on guiding me now also. And this is the financial support I get from different funding agency in doing that. And thank you. If you have any question, please.
Thank you so much, Dr. Samrat, for a nice and wonderful presentation. Let us check out some queries if there. Ruata, can you read out? I don't have it in my machine. Ruata,
Yeah, yes, sir.
can you read out the questions?
Yeah, currently they have not posted any questions, sir.
Okay.
I hope I'm not too fast. I tried to call
No, you are on time. No. Dr. Samrat, though I was switching machines and some other official assignment also could not attend completely. But I know you are using, I think, some data analytics and integrating some machine learning, is it?
No, it's not machine learning. It's ODE-based models only.
Okay,
So graph theory.
Can you just highlight it a little bit?
Sorry.
Yeah, please go ahead and highlight a little bit more of that, data, how you.
Okay. No, it's mainly, see, the ODE-based models, we normally, like in ecological and all, we go for 2-3 dimension things. So that the same things we are now doing for a larger scale. Suppose 2000 variables you have, and then you have 5000 parameters. Then it's not possible to do it analytically in that sense, and we take help for the computer. But the more or less the concept is same. We go for the steady state there, but it's more on computational part. So two main things are there. Actually, one is the graph theory which is used on other statistical things that to analyze the data. But actually to know the mechanism or the dynamical changes happening there, you actually have to use that model, which is the strength of the model. Any mathematical model gives you that dynamical changes happening there. So the idea of these things, that is exactly what I have tried to show you, or the talk is about, that you can actually use the mathematical model and the big data analysis combined together will give you a lot of fascinating results. So big data is actually the real data you are taking from there. You are obtaining some information from there. But then you use mathematical model to go beyond what is actually shown in the data, to give some hypotheses, some prediction, which can go for the experimental data. So this is something which we are trying here.
So do you actually estimate those parameters from the data?
In some cases, yes, of course. In some cases, for example, it goes two ways. If we are making the model directly from the data, so in that case, so we don't need to estimate, because what is happening, we are knowing it. If we are making the model and then going back to the data, then in that case, we need to estimate. So
right.
It depends on that.
So let me thank you once again, and I hope participants benefited. Maybe they can communicate and contact you also.
Yes, sure. They can
whenever needed.
Sure, they can always.
Look forward to future collaboration, and
Sure.
that will improve our own research also.
No, me also. It's always learning. When you see this, when people ask question, or in future if they this will of course increase, because something this is I'm also learning. So I have switched from the epidemiological and ecological things which I've learned from sir. Now I'm just switching to a different field. So it's a learning process for me also.
Yes.
And so let's see how it goes.
Yeah, that's great. That's how even research areas also they go from higher to higher levels.
No, no, sure. If you have any query or anything, please contact me, or I will also contact them.
Sure. Thank you so much.
Thank you.
See you again.
Hello.
Okay, for other participants, next lecture, as you know, as per schedule, it will be at 2:30. So till then, let us have a break. Thank you all for joining. Thank you, Dr. Samrat.
Thank you.